right here. Yeah.
Live stream.
Yep.
And do the introduction. Let's see. Let's turn on a little bit of lights in the back so that people fall asleep. Yeah, everyone knows about the GSI symposium coming up. Stefan is one of the speakers. Please attend, and there'll be a lot of other vendors there in addition to scientific talks, posters. Spread the word on GSI events and technology seminars like this, as well as if any of you have a speaker you'd like to invite for the seminar series. We have a lot of openings in the spring, so we're looking for people to invite speakers for that series. Contact the other co-directors that are not here. Ala and Rachel will be helping out, and Yuri will introduce Illumina. There are probably some people, stragglers, are coming in a little bit later.
Right, good afternoon, everybody. It's my pleasure to introduce our today's speaker, Ryan Hegarty, who is Senior Sequencing Specialist with Illumina for the last 10 years at least. And first of all, I wanted to use this opportunity to express gratitude to this company. This is one of the most supportive companies to our sequencing core, to BU in general, and to GSI. On multiple occasions, they donated pretty expensive library kits, which enabled a lot of pilot projects for GSI. Well, you might know this company as a company that dominates next generation sequencing in the last at least 10 years, but it was not always like that. In the beginning, it was a very tough competition with pretty large companies such as Applied Biosystem, who was behind the first genome project, and Roche, which is a very big company, and it had almost 2 years advantage on the market since they developed their first sequencer. And still, Illumina outcompeted all this and other later competitors such as Thermo Fisher, so that probably if you're reading a paper on sequencing, it is a big chance that it is done with Illumina. Recently, though, next generation sequencing became interesting again because some of their patents expired, and there is a whole slew of newcomers who are trying to compete again. But in my mind, Illumina response to this competition was pretty great, and they have a new chemistry, a bunch of new instruments, and they're also investing in new technology such as single cell and proteomics. So I'm hoping to hear about all that in detail today from Ryan. And also Frank Gwynn is our account specialist. He is here if you have any questions.
Thanks for coming. Appreciate it. Hope you enjoy lunch. Sign it. Yeah. That's all I got.
Well, thank you, Yuri, for the introduction. I'm fortunate enough to cover Boston, Cambridge, and to actually see what Yuri was talking about.
You need a microphone? Yeah, there's. All right, let's set this up. Microphone.
Oh.
Probably also.
Yeah, everyone try to sit up closer. There's plenty of seating up front here.
Hello? That better?
Yes.
Awesome. Great. Well, thanks again, Yuri, for having us. Again, we're really excited to be here talking to you guys about Illumina technology, as Yuri's alluded to. There's been a lot of developments in the sequencing world over the last few years, and we want to make sure that we're out there talking to customers and researchers like yourselves, hearing about what you guys need from companies like Illumina, and really to kind of talk about how we're trying to help advance genomic-based technologies. And this talk is, moving beyond the genome. And the reason I'm talking about this is because Illumina, like I mentioned, has kind of gone through a bit of a change over the last few years, and we're really focusing on helping to, enable omic methods beyond just looking at human whole genomes. And so we'll talk today a bit about what that means, as Yuri alluded to with the single cell and some of our subsequent improvements. So today's talk, going a little delay, I'll try to get through this, have quite a bit of slides. But I want to talk briefly about Illumina innovation and what that means in today's context, how we look at things. The XLEAP and then our newest instrument, which was released 2 weeks ago. We're pretty excited about what this is going to potentially mean down the line. But then also, Yuri had asked to really focus on the PIP-seq from Fluent. So Illumina acquired a company called Fluent. They're actually based in Watertown. We acquired them in the middle of the summer, and it's actually going to help enable lower cost single cell sequencing for 3' gene expression. And finally, this is the last one here, protein prep, is to really kind of think beyond RNA work that people are traditionally doing. They're just cell-based. So just kind of get you to think differently about, maybe how you can include proteomics into your workflows. So in general, next generation sequencing has become ubiquitous throughout the entire genomics community. So helping to enable different modalities and different approaches to analyze the genome, transcriptome, proteome, even metabolome. And Illumina is always engaged with all these companies to be able to provide relationships to enable these solutions. And that hasn't changed given our acquisitions. We still work closely with these companies. But Illumina, of course, we want to continue to innovate. We can't continue to stay stagnant in our technology, and we want to increase our new offerings. This is a slide that actually Yuri first saw as well at the ABRF meeting back in April. And this is presented by our Chief Technology Officer. And this is how we approach thinking in terms of innovation with our technology. We look at, for the sequencing components themselves, we look at what we call these vectors, which is looking at the pattern flow cells. Look, how can we, increase the yield on our flow cells, but also while increasing yield, improving quality, but then also trying to reduce price. We realize, obviously, at the end of the day, price is a huge factor in how people consider their research experiments. But then beyond that, we also like to think about entire workflows and operations within the lab. It's not just the sequencing part, it's how do you store reagents, how are the reagents shipped, how is data going to be accessed afterwards. So enabling an entire workflow to allow you to get up and going and enable your research with minimal effort. And then expanding our content, which brings us over here to the right-hand side of the slide. There's a lot of development that's going to be released over the next few years with Illumina technology. Some you'll hear more probably even at ASHG coming out of that meeting, which is in the next few weeks. Some of these, of course, are working on the transcriptome, so RNA further, targeted panels. Not CRISPR-based, unfortunately, but more, say, on the oncology side. Epigenome. Epigenomics, there's a bunch of really great companies out there that do, methylation for bulk samples. Biomodal, for example, is one of them. NEB has great methylation kits. We're working on a PCR-free method that allows you to look at the 5-base genome on the instruments. So that will be more technology or methods that you'll start hearing about over the next 6 months. We're actually really excited about this because it'll allow you to truly call variants within methylated sites. The expanded genome. I know I said I was going to talk about human whole genomes, but that's still the end goal with Illumina as well, especially in the clinic, is to get people to have their whole genome sequenced and be able to understand, especially rare undiagnosed disease. And with this expanded genome, this is going to be more dedicated to our larger throughput instrument, the X, that will do library prep on the instrument and actually do mapping and what we consider our long read method, all merged into basically one lane. So we're really excited to see what that looks like. And finally, proteome. I have a bunch of slides about that as well, so Illumina protein prep. And then, of course, multi-omics visualization. Partech is a company that's been around actually for a very long time, even longer than Illumina as a company. I hadn't known that. I found that out only a few weeks ago. But we've acquired them to also help to improve upon this multi-omic visualization. So increasingly bringing together different modalities to give you better insight into your questions that you're asking. So given that introduction, I want to really talk briefly about these new advancements. So XLEAP has been probably the one that you may have heard the most about over the last year or so. And XLEAP is just new sequencing chemistry that is on our NextSeq 1000, 2000. It initially was seen with the NovaSeq X, which was released 2 years ago. And then now it is basically across our entire benchtop portfolio, or actually our entire portfolio. And what XLEAP is that it's improvements to the polymerase, but then also the dideoxynucleotides themselves. Dideoxynucleotides, of course, we need new fluorophores, we need new blockers, and we need new reversible terminators. So to be able to get XLEAP be faster, quicker, cheaper, we've had to come up with entirely new classes of this. So we're actually very ecstatic to see the results that have come out of this. We were able to even get Q40 data with it. Whether or not we report out on it is a whole other topic of discussion, which I'll be happy to talk with you offline about. But, we're very excited what this has enabled. And like I mentioned, we're finally now, with the release of the MiSeq i100 Plus, we're now able to actually offer this across all of our instruments. As I mentioned, 2 weeks ago, we released or announced the two instruments, the MiSeq i100 Plus, which is available now, although we're not actually shipping until February, and then the MiSeq i100. The MiSeq instrument, for those that have been in labs, run NGS, you've probably touched the MiSeq. MiSeq is incredibly successful instrument. It's been around for 12 to 14 years. It's been the last instrument that's used traditional 4-channel imaging and 4 dye chemistry. And finally, we've come up with an improvement to actually improve, again, performance, but also significantly reduce cost and turnaround time. And so these are the kits that are available. We will, of course, the 600 cycle kits, for immune profiling, for off-target gene editing assessment, for mutagenesis screens, like using Twist. These are really great kits. Are also available on the P1 and P2 flow cells on the NextSeq 1000. So, but eventually what will happen is these 50 and 100 million reads will become available later in 2025. So there's still time to see how things go here. I think the one thing that's two things I'll talk about that have really drawn a lot of interest in this instrument is turnaround time, but then also how we actually store the reagents. Turnaround time is going to be incredibly significant here. And more importantly, for those doing any sort of metagenomics or, again, immune profiling, you're going from 2+ days for a 2x300 run down to less than a day, while maintaining exceptionally high quality across the duration of your run. You can also do library QC, and this is actually going to be even longer than you're probably going to be doing. It's actually looking at the pooling of your adapters or pooling of your barcodes prior to doing a large sequencing run. But then also even targeted panels. If you're doing infectious disease, in fact, it's incredibly critical to have rapid turnaround time in assessing an outbreak as in the hospital settings for public health labs. You can now do 2x150 in 7 hours. So we're going from, again, a day, even longer, to 7 hours, and setting up multiple runs in a given day to help get your answers faster. As I mentioned, we do have a bunch of different kits that are available. As we go into the year, we can talk more about those. We're also, as Yuri said, we're very supportive of GSI. Frankie and I will be at our table at GSI in a few weeks, so always happy to answer more questions about this as well. But I think, as I mentioned, the other thing is around the storage of reagents. we've tried to become more actually more environmentally friendly over the years. You know, for those, I mean, I've been working on this technology now for 17 years, 18 years. I've seen the first boxes. I've unpacked the large box that has one little box of 5X. Like I've gone through that pain. I've gone the dry ice burns. I think we all have. But Illumina has really tried to make that effort to reduce all of that and make it a lot easier. And so with the i100, we're actually shipping everything dry. Everything's at room temperature. There's no -20, there's no -80. It's all at room temperature. And what's great actually about the i100 is that it's actually self-installable. So going from receiving the instrument to setting up a run, you could be going within an hour. So we're actually really excited. I know a lot of our researchers are really excited about this, especially those that are working in infectious disease areas where storage is a really big challenge. And as you can see here, this is what a normal, you know, Yuri's seen this with the NextSeq reagents as well as where we've also reduced our footprint. You know, we're going from all these boxes down to an 85% reduction. So again, getting up and going is a lot easier. Then finally, as I mentioned, talking about is quality. 2x300 runs are becoming, they've always been common, but I think that we're seeing more and more, especially even on the gene editing side, making sure that you're really covering those large gene insertions, any sort of any unintended structural rearrangements by using methods like Luditas, having the high quality across the duration of your run. So if you look at this, you know, the I100 and actually your NextSeq 1000 is going to look fairly similar to this as well. But you'll notice, again, that's quite a significant improvement in that quality while getting significantly more runs over time. So obviously we're really excited about that. But I know one thing that, yeah.
Will sequencing runs on the MySeq I100 reflect the library's quality also if we then sequence that same library on a NextSeq?
Yes. Yeah, so the I100 also will have, it's a pattern flow cell. So it is CMOS though, so it's a little bit different in how we capture the images and do the imaging. One of the reasons why it's so fast is that the stage doesn't move. And so we actually take an image of the entire CMOS location. And so there's no movement. So actually your quality is probably gonna look a little bit better. I will also say too is that Q score is gonna be different. Q scores are gonna be more reflective of what you see on the NextSeq. Then you might not have the same exact bins. So MySeq traditional has what we call now has high resolution Q scores, where we report, you know, Q11, 12, 13, 14, all the way up to 37. This is going to have similar Q bins to what we see on the X, where we only have 3 bins. And so there's going to be a better reflection in what you see on those higher throughput instruments.
So basically what I'm wondering is, can we use the MySeq to do a quick QC run on a library that's got 48 samples on it to see if the barcodes are all even?
Yep.
And if they're not, then readjust and then have something better to go on the NextSeq.
Yeah, no, absolutely. And that's a key workflow, actually, is we have there on board, there's a onboard analysis workflow called Library QC. And it actually will spit out a report that does look at that index distribution. And the workflow on the instrument is actually different itself, where it actually does the index reads first. That's different than what we see with our other technologies where you do, you know, template read, then your 2 indexes, and then you do second. Then on this one, it's actually the 2 indexes first, so you can get a quicker assessment. So you're talking probably like maybe hours of that to do that QC stuff. Yep. And then it spits out a report.
The colors are the same?
So that's a good question. They are the same, so it's still blue-green. What's actually going to be different is actually the way the A base is called. So with existing 2-channel chemistry, the base that shows up in both channels actually has a bit of the same dye that shows up, one in green, one in the blue. With this, there's actually a completely new fluorophore that's excitable on both channels. So it's a completely new dye, but it has that spectral capability to be called in both blue and green, which actually makes it more stable. And we're actually able to populate the base calls around basically the center of that cluster better of how we do our base calling. So it's going to be a little bit different, but still not too much. Yep. And of course, obviously, we're more than happy to have further discussions about the I-100 offline as well, or like I said, when we're here in 2 weeks. Because I really wanted to focus mainly on this fluent PIP-seq technology and workflow. Obviously, we are very excited about the I-100 Plus, but we're just as excited about this. I mean, just seeing how single cell has changed the field over the last 10 years has been amazing. What 10X has done over the years, especially with the different modalities. And you see new papers coming out every single day, how people have hijacked 10X instruments to be able to look at, methylation, or there's some that even use probes for bacterial, so they can do single cell bacterial sequencing. However, there's still a bunch of limitations that come with working with microfluidic devices. And this is where we're trying to go with fluent PIP-seq. And of course, for those that have, it still actually amazes me, we even have lots of customers that we still have to explain the basics of next generation sequencing. And so we still assume the same too with single cell and why people might use single cell. And what we see here obviously is understanding development of complex diseases or rearrangements, potentially looking at RNA velocity, how do cells change over time, looking at their transcription patterns, but then also detecting how different cell populations are how they react to different external factors. And of course, single cell screening for CRISPR is massive. Perturb-seq, crop-seq, you name it. I mean, these are just methods that seems like everybody's doing to investigate, functional pathways or immune responses. And there are, of course, hosts of different modalities to look at. Right now, of course, we don't offer DNA-seq, and actually the offering with PIP-seq is going to be fairly limited to what you might see. So it's not going to be able to answer all your questions. However, we still have all these amazing technologies out there. BioScribe has been actually really great with looking at single cells for gene editing, give it a little bit of a higher resolution there. Mission Bio as well. But as you can see, 10X, they're still by far I would actually still say the leader in this field right now. They're just incredibly popular. But again, the technology does come with some challenges, and we hope that we can address that with PIP-seq. And as I mentioned, Illumina acquired this company called Fluent. And PIP-seq is actually a technology that's called particle templated instant partitions. Still always kind of makes me struggle. I still keep on struggling to see what that actually means. But the idea here is that it's instrument-free single cell preparation using basically using emulsion type workflows to be able to capture single cells and be able to do the entire workflow manually without relying on microfluidic devices. And so there are a whole bunch of many benefits to this kit and workflow. And I'd say one of the main advantages here is actually getting started. I know 10X did release a new small throughput instrument that's focused on just 3' gene expression, but that still can be fairly expensive. With this, you just make one purchase of these two components here. This is about $3,000 to get you started, and this is all you need. Then whatever throughput kit you're needing to look at it, to look for your experiment or how many cells you're looking at. And so this kit is just a hot water bath and then a specific vortexer. don't try to use your own vortexer. This has been calibrated specifically to enable the oil droplets and the cells to mix together properly within the tubes that you'll order, depending on your throughput. And then it again comes down to whatever throughput you're looking at. And this is where I think that the workflow is going to be exceptionally beneficial for multiple different types of researchers. You don't have to wait until you have a massive amount of cells to do an experiment. Or, you know, say if you're waiting for another researcher, you need to fill up an 8-channel, you know, kit, flow cell, or whatever. You don't need to wait for that to happen. You can go and process these individually in tubes. So we have all these different workflows from the T2, so the T means 1,000. So all the way down to 2,000 cells of capture, all the way up to about 1 million cells. So we're currently working on this as well, to be able to get to those really high throughput experiment cells. Say if you're doing genome-wide perturbation screens on immunology, that's where you'd start to really look at these high throughput. And as you've probably seen in the news, there's a bunch of companies that are really looking at that 1 million or 1 million or even up to multiple millions of cells within a given experiment. But you know, with this, our workflow, we do have the ability to look at cell surface proteins. So we also have C-seq, which is this is a component that allows you to actually do more crop-seq-like application for gene editing. And of course, across all different types of input, cells, nuclei, fresh fix, plants and animals. And I think that that's really going to be one of the key aspects of this technology as well, is the fact that you can actually tune the size of these pips to capture different size cells. So as you can imagine, when you're working with a microfluidic device, you could end up clogging those depending if you have larger cells like astrocytes or neurons. They're very large, they're very difficult to work with. But with having a larger pip, you're actually able to capture these different types of cells with a little bit more ease. And for that, Fluent does actually have a whole bunch of demonstrated applications across different input material, and this is just some of it. I mean, obviously, you know, if that's something that you're interested in, please let us know. We can still work with the Fluent part of the acquisition. The 2 of the founders are still working with us. So they have obviously a bit more experience than us Illumina people about what has been done. So please let us know if there's anything that you're maybe interested in looking at. But to get into it a little bit more is to describe how PIP-seq work. As I mentioned, we're actually looking at these emulsifications to be able to generate, and this is actually what the PIPs look like under the microscope. This is about 35 microliters. You get about 4,000 different cells for input. And what you do here is that you have your barcoded templates, lysis reagents, and single cells. They're all added to a tube. You then add in that oil on top of it, and then you actually cause that interaction for vortexing by 2 minutes. And it really is as simple as that interaction between the template or the beads and the oil themselves. And what's actually great about this, though, is that we obviously put in way too many of these pips so that you don't have to worry about doublet rates. Doublet rates are something on the rate of about 5%. Because again, you're really just reducing the chances that 2 cells will actually go into that same pip. So then after we do this, we actually then trigger lysis and then do mRNA capture. And that is essentially what you end up doing at the benchtop. And as I mentioned, you know, if you are looking at different types of cells or different types of throughput, you know, workflows, you can do anywhere from 200 cells from sorting. We do see a lot of organoids have been huge. Seeing more and more organoids. In fact, there's actually a really good talk at the Northeast Regional Core Directors meeting where they talked about there's a company called Neurocell where they actually are able to create much better brain organoids because of their growth media that they use. And it's really interesting that a lot of people use single cell for that. But really, your scaling up is just really depending on what kit and then the tube that you use. And then that's it. So this is the workflow. It's about 2.5 days. I will say is that, of course, if you're used to 10x, you know, you're going to have to be the 10x machine. It's not always going to be pretty. I'll tell you too, one of the reasons actually back in the day why Illumina 102 over 454 was the emulsification process to create libraries. I was part of a team that was looking at the technologies, and back in that day you had to break emulsions to create libraries. And when you're trying to scale up, that's not always fun. But here, again, we're looking at thousands or hundreds of thousands of cells together in that one tube. So this is all done in parallel. But I really want to draw your attention really to two steps that differentiate this. One is a stop step. So this is actually really key if you're doing time series. So say if you want to do 12 hours, you want to do 24 hours, whatever, you can keep these after this RNA capture step for up to 96 hours. So then you can then go into process everything else together. So you're able to do that, reduce a lot of variability in your processing. The second is actually this unique limited cycle PCR in creation of what we call IMIs. So these are going to be different than UMIs. And I'll actually go into explain what that is in a next slide or two. But then you do all that and then ligation and then go into your DNA sequencing. Yep.
The RNA capture step,
Yes.
is that still 3' end bias? Like with
Yes. Yep. Yeah, so what's different here is that we do have a partial, I guess, adapter sequence as part of that capture, but it is still more poly-A based, as you can see here. We do have the similar, cell barcode, as well as these spacers, but the capture so this is actually version 4.0 plus has been on the market for a little while. Version 5 is the newest version. There's actually a bioRxiv paper that came out over the summer that talks about this a bit too. So this is actually going to be a little bit different, but it still is relying on more poly-A type workflows.
Is there a version that could be 5' end capture?
We're working on it. Yeah. So that's I think one of the reasons why Illumina actually purchased it is that we can actually dedicate more development resources to actually improve upon our offerings. Because obviously, as you can imagine, one method that is constantly asked about whenever we're doing 3' is ATAC-seq. And just right now it's not feasible. And so we're dedicating more resources to be able to do ATAC-seq, do multi-omic type workflows. 5' VDJ work as well is certainly stuff that's in the roadmap. So right now, that's where we're really focusing on right now is the 3' expression, as well as the more crop-seq and actually perturb-seq. We'll be having a kit that's more dedicated to that early next year. We actually had a release note, a press release that we're collaborating with LeBron for a very high throughput experiment. They're working with them for the perturb workflow. Okay. But this is, again, our workflow, how we capture. But one thing about UMIs is that they can actually be, there's proprietary information around them. So for those that have been in the industry, one thing that has worn down, I think, a lot of scientists in the industry as well is litigation between companies. There's been a lot of people suing each other, saying that we're infringing, but so one of the ways that, not me as an Illumina, but there's just a lot of litigation going on. And one way to go around that and make sure that we can offer this is by changing the way that we call the unique fragments. And that's what we do by introducing IMIs. With UMIs, I think one of the biggest challenges is that this is actually added during the capture step, during its pre-PCR. So there's certainly challenges. You could end up introducing errors in the UMI counts and therefore decreasing the amount of, say, unique templates that are present. And so one way that we actually get around this is by actually introducing or doing an enzymatic fragmentation with a limited PCR to add in these IMI fragments at the end of our constructs. And so what this is just basically a way it's actually a analysis software that's able to, between the bases and the insertion, the IMI that's added, as well as the fragment length, we're actually able to accurately or recover more transcripts than what we had seen with just using UMIs. More importantly though, this is again actually after or post capture, post piece and around the more creating that final library step. So we're not introducing any sort of errors at the beginning of our workflow to capture these transcripts.
You probably said this, but what does it stand for?
Particle.
No, IMI.
Oh, IMI. That's a good question. I don't have this in here.
Illumina.
Yeah, we love our we love our TLAs. But yeah, that's a good question. I'll get to that. It's actually in the paper.
Does it stand for Illumina?
Yeah, surprisingly not now. But yeah, I can find that out for you.
Ryan, as you advance to the next slide, I think it's good. I just wanted to ask, has anyone here used Fluent before? Any of the Fluent for previous versions?
We did early on, and
You guys use it, yeah?
it's very scalable. When we did it,
Scalable,
it
the
was
results
only
were like
1,000
quality,
cells.
iffy. Were there any issues? I just wanted to re-emphasize a point that we keep hearing internally, and I might be speaking a little bit out of turn, I guess, but is that the PIP-seq V, the improved sensitivity, I think that was part of the reason why Fluent acquisition was such a big thing for Illumina, and why they went for it specifically and put more development into this version of the chemistry, this V5. Because what we hear in conversations over and over again, I know at Nerds last week, it was like, we'll use the past version of the chemistry and quality may have been iffy or it wasn't great, or they've seen some publications about it and it wasn't ideal, but what our technical specialists, internal and external, keep saying over and over again is this different? And let's just get it in everyone's hands to give it a try. So it's pretty exciting to be, yeah, sorry. I just
Is
wanted
the
to-
cost a fraction of a 10x run?
Yes.
Yeah. So,
Yeah, I'll show a slide that
Yeah.
actually covers that cost.
And because what I'm intrigued in trying is we've done a 10x run on mosquito cells infected with dengue virus, where we see which cells are infected, which are not.
Yep.
And might be interested in trying this again with Fluent instead and seeing how that data compares to a 10x run.
Yeah, absolutely. And we can certainly have a follow-up conversation.
And the number that, and I know you have a slide on it, but the number that comes to my head from conversations last week is a 2,000 cell capture, like an 8-pack, basically comes down to for $300 per sample-ish list price, right?
Yeah.
So $300 for a 2,000 cell capture, which is pretty competitive, stacking up to the 10x, right? Especially when you think about the quality of the cells you're capturing, the lower delta rates. Yeah.
Frankie, you're stealing some of my slides.
Oh, sorry.
But Yuri, you did say scaling is a challenge with this.
No, they said it's very scalable.
Oh, right.
And it looks like it is, because back then it was 1,000 cells, and now it's 100,000, maybe a million. So.
Yep. All right, I just was thinking in terms of like multiple samples, I guess, would be certainly a challenge. And I actually won't talk about this, but we do allow for cell hashing. So it is 5 cents per cell, the lowest point. And that's before cell hashing. So you can imagine that you can get down further the cost per cell. But yes. But yeah, the scalability is there. It's just a matter of like how many can you process per one operator, I think. But obviously that's where we're also trying to work internally, bring the development is on potentially automation as well down the line. But as we can see, and I will also say, unfortunately, is that about the same, basically within the same week that that Illumina acquired Fluent, there was actually a benchmarking study that came out of California. I think it was Genentech did a benchmarking study on a bunch of 3' stuff, and it didn't look good for the PIP-seq, but it was using V3. So if you come across that paper, please keep that in mind. That's an older technology that is not on the market right now. But as you can see with the V or version 5 chemistry, we're actually able to, increase the amount of transcripts per cell. This is a very busy slide. Basically, a lot of the specs that you should expect from these, again, they're based off of PBMCs, so it's cheating a little bit, but it's how everybody benchmarks. Really what we just want to make sure to get across, up to 85% capture efficiency, multiplet rate less than 5%. Ambient RNA also is not an issue as well. Again, because you're just basically colliding your cells with these droplets. Ambient RNA should not be captured. So that can certainly be a challenge with other methods as well. And as I mentioned, comparing against older technology, we go and we do this on this slide. We're comparing against the 10xV 3.1. But what we really want to kind of show here is that with version 5 is that we're able to reproduce a lot of what can be done with existing 10x, but then actually improve upon it by getting unique cell types. So these dendritic cells, HSPCs, these are cells that might be difficult to capture within the 10x because of the microfluidic limitations. And we're actually able to capture those using a non-device oriented workflow. Oops, what did I do here?
So just a quick question. You use a vortexer to quickly mix the suspension of cells with the emulsion.
Yep.
And that vortexing rate is gentle enough that we won't rupture cells?
Exactly. Yep. And that's why you need to specifically use that vortex.
Okay.
So that you don't disrupt the cells.
Does that work with nuclei as well?
Yes. Yep. So these are just some of the examples of how people have used single cell sequencing using the T2. I think one of the greatest uses of this is that it actually drives the ability to teach people how to do single cell without incurring massive costs. In this case, this is actually at a school in Brooklyn where they had students prepping single cell at the high school level at about $300 per tube. So if there's any sort of public outreach where you really want to teach kids about biotech and single cell and want to get some hands-on time, that's a capability. I think also if there's interest in doing this across a broader BU community, we could also probably do like an on-site day where we actually do more kind of a hands-on training down the line. And Frankie and I are, the FAS who is working for Fluent is based locally, so more than happy to try to set something up and work with, Yuri in the core. For the T20, they're actually able to do larger studies, but then also in each of these studies able to find rare cell types that they weren't able to, again, find within 10x because of microfluidic limitations. And then finally with the T100, looking at neuroscience. In this case, that they were looking at astrocytes that were only present at 0.1% of the cell population. And these are notoriously difficult to capture, and they were actually able to capture them using PIPSI. So as you can see, using the core, with Yuri and the NextSeqs that he has, you have that ability to do any sort of size experimental workflow for a single cell using Fluent. Anywhere from the P1 flow cell for your smaller cell sorting, all the way up to the P4 and these larger experiments. With the idea too is that eventually, obviously we'd appreciate for you to use our analysis software, but we also realize, you also have your own options, but we do have the solution for using Partech and Illumina as well.
Yeah, so we've heard of that company before. You guys bought it.
Yeah. So Illumina is very dedicated to create end-to-end workflows. It's centered around Dragon, which is our FPGA hardware-software solution, which is on the human genome side has been used for the All of Us project, Genome England, but it's also being used for a lot of other larger single-cell studies as well. That's the foundation for doing a lot of the genes cell by gene matrices, everything like that. But then also Partech is going to be a way to visualize all your multi-omic modalities together. So just briefly, yeah,
They used to work as a subscription model.
yeah. So what's going to happen, I believe, with this is that when you get a PIP-C kit, that you actually get access to this. And obviously we'll talk about it more. So Fluent is still, we're still integrating Fluent and Partech into the Illumina environment. And I'll actually talk about that. I'm gonna go ahead in one slide, but basically we're gonna start to have an Illumina branded single cell workflow coming out beginning of 2025.
So we looked at Partech a couple of years ago, and it was limited to mouse and human.
Yeah.
Is it now adaptable to any organism you want, or any with the genome?
That's a good question. I think that we're still figuring that out at the moment, how it's being integrated. I want to say that we're going to enable flexibility to be able to look at beyond, the two model organisms, but I think it's a bit early to say at the moment.
Okay.
I'm not. So we actually have an informatics specialist, Dan Giba, who presents on a lot of these topics. He'll actually be in town the week of GSI.
Yeah.
If he's here and he has availability, we could see about having a meeting, or we can certainly just set up a call too. Our informatics solutions have gone through the roof recently. We've put a lot of effort into solutions. So Dragon specifically, what I've always found interesting about Dragon was that it's founded by the same people that actually not founded, but they worked on credit card transactions. So it's actually, you can imagine how fast you need a transaction to happen when you swipe your card. You just take it for granted, right? It's like, a second, whatever. The people that designed Dragon actually were part of that credit card transaction field.
Not sure if that instills confidence.
We're not swiping your data,
right.
genetic or financial.
Okay.
But I think it's just really interesting. Actually, the guy that developed the Picard pipeline, he came from banking. He was in.
But those are very rigid systems. What we're looking for is flexibility, because we have a lot of different, systems, species. I know mouse and human is still heavy, but we also have Coelians, Drosophila, zebrafish.
Yeah. Yeah. No, and we recognize that. I think it's just. I think what ends up being challenging, I think, at Illumina, quite frankly, is resources. Being able to dedicate that to be able to bring all this information in and make it an end-to-end solution that our customers can use. But we're always open to discussions and hearing feedback. So I kind of talk about this briefly again when we're looking at the potential cost per cell. Again, when we're looking at Flex is a little bit more relevant here, but we're getting that cost down to a few cents per cell. And as I alluded to, sample prep through analysis, this will be an end-to-end workflow branded under the Illumina name come 2025. So that's PIP-C. I think we're really excited to bring it on and see what you guys do with it. Obviously, if there's anything that we can help with, test it out or give it a run through, let Frankie and I know. I'm more than happy to work with you guys. So that being said, I'm going to go completely nuclear here and go at a completely larger scale. But I think it's that final omic methodology that is being adopted by next generation sequencing, and that's proteomics. Proteomics, of course, mass spec and gel electrophoresis, they're not going anywhere. They've been around for a long time, and they're still really the gold standard. However, they do have some limitations. There's scale, there's limited detection, high sample input quality. And so what we've actually seen over the last few years, mainly with a company called Olink, they came out of Sweden, Uppsala, and they actually have a headquarters here in Watertown, is actually being able to look at, you know, proteins at a massive scale beyond the limitations of what you see with mass spec. And I know I'm not a proteomics guy, I did it in my master's, but I'm not exactly as adept at talking about it, but we have seen too is that mass spec has started to catch up on the throughput aspect of things. But what we're trying to do here is to utilize that ability of NGS to look at basically counting the presence of proteins within solutions. And that's done by either using aptamers, which is what Illumina is using, as well as antibodies using Olink. And what they actually provide is a really high affinity. There's that higher dynamic range, so we're talking significant dynamic range difference. And eventually the ease of workflow. But like I said, Illumina protein prep and this is not a replacement for mass spec. What this is a large-scale discovery platform that can help you find if proteins are present for, say, a given disease or whatever, see what's present, and then potentially validating using mass spec, using gel electrophoresis, all your traditional methods. So as protein prep itself is a streamlined solution that can go from you know, hundreds or so samples all the way through data analysis. Right now it's in early access with a few sites that have started to run this. We're looking at 6,000 proteins from either plasma or serum. With the idea, with the full commercial release at the beginning of next year for anywhere from 9,000 to 11,000 proteins detectable. What this is a collaboration between us and two other companies. One is SomaLogic, a company formerly called SomaLogic. They're now part of Standard BioTools. And then also we have TCAN. TCAN provides the automation. And this is a TCAN instrument that's dedicated solely to this workflow because of all the special instrumentation that you need to be able to have these aptamers bind to your proteins. And as you know, dedicated right now to more high throughput instruments like the X or the 6,000, with the goal that eventually it will be on the 2,000, especially the P4 flow cell next year. And how do we actually capture these? So we use what are called SOMAmers. So these are slow off-rate modified aptamers. Aptamers are, you know, synthetic strands of DNA that have modified nucleotides. And what they can do is that they're able to bind within unique motifs of proteins themselves. And so it has a unique sequence that can find these specific motifs. And then on the other side, it actually has a small little barcode that you then PCR off to then be able to then amplify that out and then count if that protein is present or not. And these are designed against specific targets, and they're, again, very difficult to remove from that binding once it's actually done. So once it's bound, we then capture the protein and the tag itself, and then enrich to then say, is this protein present? And then give the idea of the dynamic range of that protein presence. And we take into consideration all different types of pathways, anywhere from cancer, neurology, or inflammation. And where we offer it is that we're going to have a much larger offering than what's currently out there with Olink and their antibody solution. One key thing about aptamers, of course, is that they are more easily reproducible than the antibodies. One thing that is difficult with antibodies is that you have to keep on going through reproducing them through animal models, especially monoclonals. And what you have to do to be able to go from batch to batch is actually do a bridging step where you have to sequence a bunch of samples to then make sure that the antibodies are similar between each batch. With aptamers, that's not necessary. Again, you're just synthesizing fragments. And we validate these in many different ways for orthogonal validation using more rigorous methods than what are existing out there. And so what we're able to do by doing this is have increased reproducibility in between batches. and allowing us to get down to that femtomolar sensitivity. So again, we're looking at very low amounts of protein within your given serum sample or plasma sample. Eventually, we'll move on to multiple other types of input, including cell lysates. Hopefully, cell lysates is out next year, but that's going to be the goal. And of course, we need to have low variability. One thing that's great about this, so this has been in early access at a bunch of different locations. We have pooled samples together, then looked at that percent CV variation between plate to plate and site to site over a course of runs, and our CV is close to 7 or 8%. Whereas you look at an antibody type workflow that uses smaller volumes, that's somewhere around in the 30s. You can't have that when you're looking at detecting presence of very small proteins or low concentration proteins. So we're really excited to see how this has worked out. TGen is one of our early access sites. They've been running this, and we're just looking at different, protein presence within, say an inflammatory response. So this is the control. These are all your different samples. Looking at cancer, you expect a bunch of different inflammatory signals. So you can see how that compares against the control, as well as looking at fatty acid binding proteins. You can see with the control, these are the amount of counts you have, but then in this cancer type, it's significantly higher. So this could be something that you can look at what proteins are different here. So this is, again, early access. Talk to us if there's anything that you're interested in running. We can help set up with one of the other sites to do a test, see what that looks like. But again, we're really excited to see how proteins are beyond just cell surface proteins that are done at the single cell level are looked at using next generation sequencing. With all that being said, of course, we realize that there's a lot of information out there. I mean, there's seemingly a new method published every 2 days, but I always like to say by the time I finish giving a talk, there's a new method published and I'm already out of date. But we do have a nice resource online that will, for if say you're new to NGS or somebody that you're in a lab with needs some information. We have this new iResources that's available for core labs. Just gives you a whole bunch of different new material.
Excuse me.
Yeah.
For the proteins.
Yeah.
Does the platform provide resolution for different types of forms?
Yes, for different types of forms, but not for post-translational modifications. We can't get specific modification changes just yet, but it will allow for different isoforms.
Are the aptamers recognizing the peptide's amino acid sequence or a structure?
Sequence.
Sequence?
Yeah.
So you do denature your protein lysate?
I'm not positive on that. I'd have to see what the protocol looks like, but I'm pretty sure we do. Yeah, I'm not too sure on that, honestly. Like I said, this is still relatively early access. We've actually been talking about it for 2 years, but it's only finally come into fruition. But it's a good question. It's something that we can follow up on as well. All that being said, as Yuri mentioned, we take our relationships with our customers seriously, with our academic researchers. You guys are what help drive drive research. We want to help enable you as much as possible. We have all different options that are out there. Please let us know if there's any way that Frankie, myself, Yuri, that we can help you with your research and get you going. So that being said, thank you for your time. I think we're right at time, so if you want to, I'll stay around if there are any questions you want to ask. If not, then too, if you think of any, as I said, we'll be at the GSI in 2 weeks, so you can ask then.
We should have Fluent with us there as well.
Yes, Fluent will be there as well.
Representation.
Yep, so thank you.
Thanks, everybody.
Thank you.
workshop.
The
cost is scalable.
Yes.
Some protein, like antibody against the protein, so
So it's actually a sequence. So we have modified
Pilots are really
nucleotides within the application.